of our quiz, which is multiple choice on Chapter 18. Shouldn't take very long, and then we're done with all the new lecture material. Huh?
Are we doing the...
This is... Huh? For the quiz? The quiz is organic. That was the last chapter. That was the last exam. Yeah. I'm pretty sure the quiz is supposed to be organic chemistry. Yes, it is. Okay.
Yeah.
Like, I hope I wrote what's on the syllabus. It wouldn't make sense otherwise. All right, so here we go on biochem. Biochem basically it's one of the more interesting sections because this applies to our lives. We're talking about chemistry in our bodies here. So, for those of you who have taken biology recently, a lot of this might actually be review. If you've taken physiology at all, I think you have to chemistry before you do that, though. But it's a lot more biological in nature than it is actual chemistry. So kind of a just quick review here about the cell. When we're looking at biochemistry, we're looking at stuff on the cellular level. So we need to understand exactly the cell, and it's just, like I said, applied through in biochem. So the cell is the smallest structural unit of living organisms that has the properties traditionally associated with life. And it can be an independent living organism or a building block of more complex organisms. So we have singular cellular organisms, and then we have multiple cellular ones like us. Some cells contain a nucleus, and that's the part of the cell that contains the genetic materials. Others don't. The perimeter of the cell is bound by a cell membrane that holds the contents of the cell together. And then the region between the nucleus and the cell membrane is called the cytoplasm. The cytoplasm contains a number of specialized structures that carry out much of the cell's work. We're not going into the difference between plant cells and animal cells in here. We don't care that much, but this is just kind of our overview of the cell. So here's our cell, the nice picture of it. The primary genetic material, remember, is stored in the nucleus in the very center. We have our cell membrane on the outside, and our cytoplasm is in the inside with our ribosomes and chromosomes and all that other stuff that's in there. The main chemical components of the cell can be divided into 4 classes. We have carbohydrates, lipids, proteins, and nucleic acids. So we're going to divide these into our 4 components and look at each of those components on the chemical level. The first component is carbohydrates. These are sugars, starches, and fibers. Carbohydrates are the primary molecules responsible for short-term energy. So carbs are your energy storage. This is probably something that you've heard of because we all know if you're going to go run a marathon, you need to load up on carbs. That's energy. Carbohydrates form the main structural components of plants. And they have the generic formula of CH2O to the N on the outside. Carbohydrates are aldehydes. Remember, when we talk about an aldehyde, what is it that functional group? What is the functional group for an aldehyde? Anyone remember?
And a triple bond? Wait.
We have,
like...
RC.
What's the functional group associated with aldehyde? We have RC. There's 2 other things.
H-O with a double bond?
Yeah, exactly. So that's our aldehyde. So carbohydrates, at the end, they will have this aldehyde functional group. Along with that, sometimes all over in the middle of this, it's going to be a long chain, and they'll have lots of alcohol groups attached to it as well, so those OH groups. Sometimes they're also ketones. What was the difference between an aldehyde and a ketone?
It's instead of the H, it's the R, another R.
Yeah. So for the ketone, you might have a chain on this side, and you're going to have a chain on that side that can either be the same or a different chain. Okay, so here is glucose, fructose, and galactose as examples of our carbohydrates. This formula is for glucose. So we see up at the top, we have our aldehyde group at the very top. Then you see this chain of carbons, and you have OH groups, the alcohol groups, down the side of the chain. Glucose is the aldehyde, like I said, and the many OH groups on it makes glucose soluble in water and blood. This is important because if you didn't have it soluble in blood, it wouldn't mix in with our blood in our bodies, and we couldn't get our energy. Glucose is easily transported in the bloodstream and is soluble within the aqueous interior of the cell. So remember, we're looking at hydrocarbon chains. Hydrocarbon chains typically are nonpolar. So those OH groups now add the ability to have hydrogen bonding. They increase the polarity there. Glucose is an example of a monosaccharide, which is a carbohydrate that cannot be broken down into simpler carbohydrates. Monosaccharides such as glucose will rearrange to form a ring structure in solution. So in our bodies, glucose actually exists more like this. It doesn't exist in this straight chain. So they don't exist, these straight chain forms in our bodies. They do this rearrangement, and they form those ring structures. Glucose, fructose, and galactose are examples of hexoses. Hexose, what does hexose mean? What do you think? 6. So hexose is 6, the 6-carbon structure. What do you think of pentose is? 5-carbon structure. So use the terminology that, like I said, multiple choice test. Use those terms to kind of help get you the right answer. So glucose, fructose, and galactose are hexoses, the 6-carbon sugars. These are the most common monosaccharides in living organisms, or the most common ones are pentoses and hexoses. Fructose and galactose form rings that are isomers of glucose. So this shows you fructose right here and galactose over here. And if you notice in these, they're very similar in structure. A lot of times what will happen is you'll just have, if you look at galactose and we go back to glucose, you'll see that the difference between the two of them is only this OH group right here. On glucose, it points down. Oops, wrong way. On galactose, it points up. So sometimes it's just that one little difference it differentiates between one sugar and another. So something very simple like that. Okay, fructose is also known as fruit sugar, and it's a hexose found in many fruits and vegetables, and is a major component of honey. Galactose is a brain sugar, and it's a hexose usually found combined with other monosaccharides and substances such as lactose. And galactose is also present within the brain and nervous system of most animals. Glucose and fructose will condense to form sucrose. So we have 2 monosaccharides up at the top. We have our glucose and our fructose. These are 2 monosaccharides. They combine to form a disaccharide down here. So a disaccharide is 2 saccharides put together. So you have glucose and fructose forming sucrose. They are combined by what's called a glycosidic linkage. A glycosidic linkage is just an oxygen that connects 2 carbons of adjoining monosaccharides. So you have 2 monosaccharides, and they're combined by an oxygen, and that's a glycosidic linkage. Monosaccharides can link together as well as forming disaccharides. They will also form polysaccharides, which is just multiple. So poly is multiple. Long chain-like molecules that are composed of many monosaccharide units. Polysaccharides are a type of polymer. And these are chemical compounds composed of repeating structural units in a long chain. So starch is a form of a polysaccharide. Once again, you have a bunch of monosaccharides here that are linked together by this glycosidic linkage. So whenever you see this O in between 2 of these big ring structures, that's what's considered a glycosidic linkage. Monosaccharides and disaccharides are simple sugars or simple carbohydrates, whereas the polysaccharides are complex ones. Some common polysaccharides are starch and cellulose, both of which are composed of repeating glucose units. The difference between starch and cellulose, it's very minor. Starch, we have this glycosidic linkage points down. With cellulose, it actually goes up. So that's the only difference between those 2. A third kind of polysaccharide is glycogen. Glycogen is a structure similar to starch, but the chain is highly branched. In animals, excess glucose in the blood is stored as glycogen unit until it's needed. The difference between starch and cellulose is just the link between those glucose units. In starch, the oxygen atom joining the neighboring glucose units points down, like I just said, and this is what's referred to as an alpha linkage. In cellulose, the atoms are roughly parallel; they kind of go up, and that's what's referred to as a beta linkage. The differences between those linkages causes the differences in the properties. Starch is common in potatoes and grains. It's soft, pliable substance that we can easily chew and swallow. During digestion, those links between the individual cellulose units are broken, which allows those glucose molecules to pass through the intestinal wall and into the bloodstream. Cellulose, on the other hand, this is what we know as fiber. It's stiffer and more rigid substance. We can't actually digest it. So cellulose is the main structural component of plants, and the bonding in cellulose makes it indigestible. When we eat it, it passes right through the intestine undigested. Okay, so to kind of give you an idea as to what to expect here, like I said, it's an overview. I'm not expecting you to automatically become experts on biochemistry here. For this, we're just looking at determining which molecules are carbohydrates, and we're going to classify it as either a monosaccharide, disaccharide, or polysaccharide if it is a carbohydrate. So looking at this first one up here, what do you all think this is? Is that a carbohydrate? What does it have to be a carbohydrate? What did we say? I think it's still written on the board. Do you see an aldehyde or a ketone functional group there?
No, because there's no double bond.
There's no double bonds, right? So is that a carbohydrate? That one's not a carbohydrate. How about B?
Yeah.
Yes, that is a carbohydrate. Is that a mono or a disaccharide? Mono. How do I know it's mono? There's just one of them, right? So there's just one thing. What about C?
Yeah. There's a double bond.
There's a double bond, but is that a ketone or an aldehyde? Or, sorry, yeah, ketone or aldehyde?
No.
No. What type of functional group is this called over here? What was that?
Is that the, is it an alcohol?
It's not an alcohol.
Carboxylic acid.
Carboxylic acid, yeah. So that's not going to be a carbohydrate. And then how about D?
That's the
Disaccharide, yeah. Okay, let's see here. We're going to go on. Okay. Lipids are chemical components of the cells that are insoluble in water but soluble in nonpolar solvents. So lipids, those are the things that we know as fats. They include fatty acids, fats, oils, phospholipids, glycolipids, and steroids. Insolubility in water makes lipids an ideal structural component of cell membranes. Lipids are used for long-term energy storage and for insulation. Lipids are kind of, they're a little different because there's several different classes of them, so we have to look at each class individually. So when we look at fatty acids first, we look at fatty acids. They have a carboxylic acid group on them with a really long hydrocarbon tail. So there's some sort of hydrocarbon tail over here with a carboxylic acid group over here. This hydrocarbon tail down here is very nonpolar. This carboxylic acid group is very polar. So it has a polar end over here and a nonpolar end all the way down here. Myristic acid is an example of a saturated fatty acid. So saturated, when we talked about organic chemistry, when I said saturated, what did I mean by that?
It has a lot of hydrogen.
Has a lot of hydrogens. Does it have any double bonds in that hydrocarbon chain? No, it's all single bonds, right? So saturated fatty acids, the hydrocarbon tail is all single bonds. So it's saturated with hydrogens. So myristic acid is saturated. Its formula is CH3CH212COOH. So you see that there's basically a 13-carbon hydrocarbon chain over there attached to that carboxylic acid group. Its carbon chain has no double bonds. This occurs in butterfat and in coconut oil. Other fatty acids called monounsaturated or polyunsaturated fatty acids have one or more double bonds in their carbon chains. How many double bonds do you think a monounsaturated fatty acid has? One, right? Mono. So once again, look for those key terms. Polyunsaturated has obviously more than one. Oleic acid is an example of a monounsaturated fatty acid, and its formula is shown right here. This little colon right here, that's going to be, whoops, what just happened? That's where,
Okay.
someone's playing with me. That's where the double bond is. Oleic acid occurs in olive oil, peanut oil, and also in human fat. Okay, so here we see, this is the actual formula for myristic acid, and that's the 3D structure up there. Down here, we show the oleic acid, so you can see the effects that that double bond has in the structure of the compound. So now we have this kink in the chain right here, so you get this bend, and that's what the oleic acid actually looks like. Fatty acids differ only in their R group. The long hydrocarbon tails of those fatty acids, once again, make them insoluble in water. So you can see we go from butyric acid, which has the four carbons in it, all the way up to stearic acid, which has 18. Those are all saturated fatty acids, so all single bonds. And the sources for the saturated ones are things that are more solid at room temperature, if you think about it. Milk fat and whale oil, butter fat, coconut oil, beef fat, butter fat, and beef fat, and also butter fat for the stearic acid. The longer that the chain gets, the more likely they are to be solid at room temperature. When we get down to unsaturated fatty acids, we have our oleic acid, and notice that these all have 18 carbons in them, and what differs is just the number of double bonds. And so we have oleic acid, which has the olive oil and peanut oil. Linoleic acid is linseed oil and corn oil. And then we have linolenic acid, which is in linseed oil and corn oil as well. So these are just the things that differs between these is just the number of double bonds. So changing the number of double bonds, you change the compound entirely. Okay, questions so far? How are we doing on the fly-through? Fly-through, right? All right, so when we have another category of lipids is fats and oils, and these are actually called triglycerides. Sorry, got ahead of myself. Triglycerides are basically a compound that combines a glycerol molecule with 3 different fatty acids or 3 of the same fatty acids. So you have glycerol linked to 3 fatty acids, as you can see in that block diagram right there. So to form a triglyceride, what they do is they react glycerol with 3 different fatty acids. And what will happen is that you end up getting the H from the glycerol molecule will react with the OH on one of those fatty acids, and a water molecule leaves. So you have water breaking off, and then you have a linkage forming between this oxygen right here on that carbon and this carbon here on the fatty acid. So every time a triglyceride is made, you end up with 3 water molecules as a byproduct. And the linkages, these are ester linkages. If you think back to the organic chemistry that we were talking about, when you have CO with the C with that double bond O right there, that's considered an ester. Okay, this is tristerin. This is the main component of beef fat. And it's formed from the reaction of glycerol and 3 stearic acid molecules. So you have glycerol plus those 3 stearic acid molecules, and this actually shows you what that triglyceride looks like, this tristerin. So here's tristerin formed plus your 3 water molecules at the end. And obviously you're not expected to memorize these. All right, so now we're going to identify the triglycerides and classify each as either saturated or unsaturated. So up on letter A, is that a triglyceride? What do y'all think?
No.
No. How do we know it's not?
Because the glyceride isn't it's the upright one. It's not the present.
I don't even see glyceride up there. I mean, I don't know.
Yeah, 3 fatty
What is that? What functional group is on that one?
Yeah.
Carboxylic acid again, and there's a double bond in there, yeah. So that's not a triglyceride. What about B?
Yes.
Yes, B is a triglyceride. Pretty obvious to recognize these when you start to see them. How do we know that's a triglyceride?
Well, it's connected to.
It has that glycerol backbone, right? With the triglycerides, they're going to always have these 3 lines like this. And so it's pretty simple to, once you recognize that there's this huge compound that's going to be your triglyceride. Is that one saturated or unsaturated?
It is. Well, no, it has double bonds, so it's unsaturated.
Where are the double bonds for B?
Oh, we were looking at C.
Oh, yeah, letter B.
So it's saturated.
That one is saturated. There are no double bonds in it. How about C? That's a triglyceride, and that one is unsaturated because you can see the double bonds here in the middle, right in there.
Yes.
So we have some monounsaturated fatty acids in there. Is D a triglyceride?
No.
No, what is D? Carbohydrate. Okay. Okay, where are the triglycerides on this one? B and D. And is B saturated or unsaturated? Unsaturated. How about D? Saturated. Okay, questions on that? Okay, if the fatty acids in the triglyceride are saturated, the triglyceride is called a saturated fat and tends to be solid at room temperature. Lard and many animal fats are examples of saturated fats. If the fatty acids are unsaturated, we have an unsaturated fat, and that atom in that molecule tends to be liquid at room temperature. Canola oil, olive oil, and most other vegetable oils are examples of unsaturated fats. All right, another type of lipid is called a phospholipid. The basic structure for a phospholipid is similar to the triglyceride, except for one of the fatty acids is going to be replaced with a phosphate group down there. Hence the name, the phospholipid. Unlike a fatty acid, which is nonpolar, a phosphate group is polar and often will have another polar group attached to it. So the phospholipid has a molecule that has a polar section and a nonpolar section. So we have polar over here, nonpolar over there. Oops, I think I went the wrong way, sorry. If the structure, I always have fun with these names, phosphatidylcholine, a phospholipid found in the cell membrane, the polar part of the molecule is called hydrophilic. Hydrophilic is water-loving. The nonpolar part is hydrophobic, which means it kind of hates water, it avoids it. So here, this is our phosphate group. Right here you can see the phosphate. It has another thing attached to it, but this is our polar section. Over there, the hydrocarbon chains, those are our nonpolar sections. So here's our hydrophilic, there's our hydrophobic. Glycolipids have similar structures and properties to phospholipids. The nonpolar section of a glycolipid is composed of a fatty acid chain and a hydrocarbon chain, whereas the polar section has a sugar molecule such as glucose. Phospholipids and glycolipids are often schematically represented as a circle with two long tails. The structure of the phospholipids and glycolipids is ideal for constructing cell membranes. The polar parts that interact with the aqueous environment of the cell and the nonpolar interact with each other. Lipid bilayer membranes encapsulate cells in many cellular structures. So here we have a lipid bilayer membrane, and on the outside you'll see this polar head. That's the part that can dissolve in the blood, and it can mix with cells as well. In the interior there, you see the two tails. That is going to be the lipid portion, the nonpolar portion that doesn't like the water. Cell membranes are composed of lipid bilayers in which phospholipids or glycolipids form a double layer. In this bilayer, the polar heads of the molecule point outward and the nonpolar tails point inward. Okay, questions so far?
So the tails are the phobic and
the tails are, yeah, they are trying to get away from the water, so they're pointed inward. All right, some other lipids are steroids. Steroids are lipids that contain the four-ring structure that you see right up there at the top. Some common steroids include cholesterol, testosterone, and estrogen, so you can see the formulas for them. Notice that all of them have the same four-member ring as a central part of their structure. They might have some double bonds in different places. That one has more double bonds, but it's still that same four-member ring. Cholesterol serves many important functions throughout the body. Like phospholipids and glycolipids, cholesterol is part of cell membranes. Not all cholesterol is bad. Cholesterol serves as a precursor for the body to synthesize other steroids such as testosterone, a principal male hormone, and estrogen, a principal female hormone. Hormones are chemical messengers that regulate many body processes such as growth and metabolism, and they are secreted by specialized tissues and transported in the blood. Okay, so kind of relating this to our health, we talked a little bit about trans fats last time. Now we're going to look at triglycerides. Most of the fats and oils in our diet are triglycerides. During digestion, these are broken down into fatty acids, glycerol, monoglycerides, and diglycerides. These products will pass through the intestinal wall and then reassemble into triglycerides before they're absorbed into the blood. It's a slower process than digestion of other food types, and eating fats and oils gives a lasting feeling of fullness. The FDA recommends that fats and oils composed with less than 30% of your total caloric intake, and no more than 1/3 of those fats should be saturated fats. A diet high in saturated fats, we know, increases the risk of artery blockages. That's old news, I think. That can lead to stroke and heart attack, and monounsaturated fats may help protect against these threats. Questions? Okay, proteins. From a biochemical perspective, proteins have a very broad definition. Within living organisms, proteins do much of the work of maintaining life. Most of the chemical reactions that occur in living organisms are catalyzed or enabled by proteins. Proteins that act as catalysts are called enzymes. Without enzymes, life would be impossible. Proteins are the structural components of muscle, skin, and cartilage. And proteins transport oxygen in the blood and act as antibodies to fight disease and function as hormones to regulate metabolic processes. All right, so proteins. Looking at the chemical structure of these are going to be polymers of amino acids. So remember, polymers means you have multi-chain. Amino acids are molecules that contain an amine group, a carboxylic acid group, and an R group. So proteins have this R group down here, they have your amine group up here, and they have a carboxylic acid group over here. Where it's going to differ is going to be this R group. That's where you're going to see a lot of changes with the different types of proteins. The general structure of an amino acid is that right there, sorry. In a protein, an R group does not necessarily mean a pure alkyl group. So you could have all kinds of different things on that R group. It doesn't have to just be carbon and hydrogen. Amino acids differ from each other only in their R groups. The R groups or side chains of different amino acids can be very different chemically. We have, for example, alanine, which has a nonpolar side chain, while serine has a very polar one. You can see it has an alcohol group going on there. Aspartic acid has an acidic side chain. We have our COOH or carboxylic acid group, while lysine has a basic one. It has this NH2 right here. When amino acids are strung together to make a protein, these differences determine the structure and properties of the proteins. here are the 20 common amino acids, and once again, just notice that all of the amino acids, they all have the same backbone. What differs is that R chain down here. Some of them have, they have these rings down here. You have benzene rings, a few of them. And others, you have the amines down here. You have alcohols up here. So you have different groups attached to those amino acids. They're not all just carbons and hydrogens. Amino acids will link together because the amine end of one amino acid reacts with the carboxylic acid end of the other. So here we have, notice that it is the backbone that is reacting, not the R chain. The carboxylic acid group of one amino acid reacts with the amine group of another amino acid, and you get what's called a peptide bond. Whenever you see a bond between this carbon and a nitrogen, that means that you have that peptide bond. Water is a byproduct. So the resulting bond is a peptide bond, and the resulting molecule is called a dipeptide. There's two of them. Short chains of amino acids are called polypeptides. Functional proteins contain hundreds or even thousands of amino acids joined by peptide bonds. And we don't do this. Okay, the protein structure. This is where proteins can get kind of interesting. There's multiple parts to a protein structure. So in proteins, the amino acids, first they interact with one another, causing the protein chain to twist and fold in a very specific way. The exact shape that that protein takes depends on the types of amino acids and how the interactions, the intermolecular forces, is going to be a big player in this particular structure. Different amino acids in different sequences result in different shapes. Insulin, for example, is a protein that recognizes muscle cells because their surfaces contain insulin receptors, molecules that fit in a specific portion of the insulin protein. If insulin were a different shape, it would not latch onto insulin receptors on muscle cells and therefore would not do its job. So people that have insulin problems, a lot of times it's because they have a protein that's not formed right. The shape or conformation of proteins is crucial to their function. And there are 4 levels of protein structure analysis. We have primary, secondary, tertiary, and quaternary. So a kind of an overview of the protein structure here. The primary structure is the amino acid sequence. So when you're looking at the primary structure of a protein, you're looking at the order of the amino acids. So up at the top there, you can see the order of the amino acids. They have these 3-letter abbreviations. Those 3-letter abbreviations tell you what the amino acid is. Don't memorize those. So we have like alanine, leucine, serine, gluten. I don't even know. I forget what GLU is. But it tells you the order of the amino acids up there. The secondary structure refers to small-scale repeating patterns, and such as what we call, they have an alpha helix or a beta-pleated sheet. We'll look at that in more detail in a second. Tertiary structure is going to be basically the large-scale bends and folds of the protein. So you'll notice that for the tertiary structure, we start to see kind of this molecule that's bending and folding. And then the quaternary structure is when you have a whole bunch of proteins segments together, and that's the arrangement of the individual polypeptide chains. Where'd it go? Okay, so primary structure. Primary structure is the sequence of the amino acids in the chain. The primary structure is maintained by those peptide bonds between the individual amino acids. So this is that carbon to nitrogen bond. For example, one section of the insulin protein has this sequence that you see here. Each 3-letter abbreviation represents an amino acid. The first amino acid sequences for proteins were determined back in the 1950s, so this is kind of old science as far as that goes. Today, the amino acid sequences for thousands of proteins are known. Changes in that sequence of a protein, even a minor one, can have a devastating effect on the function of a protein. Okay, so primary structure. Some of the effects that it can have, actually, sickle cell anemia is caused by a modification in the primary structure of hemoglobin. Hemoglobin is composed of 4 protein chains, each containing 146 amino acid units. The substitution of glutamic acid for valine in just one position on 2 of these chains results in the sickle cell anemia, in which the red blood cells take on the sickle cell shape, which leads to damage of major organs. So it's basically 2 amino acids that are substituted into this huge structure caused that major defect. Secondary protein structure. There are 2 types of secondary protein structure. We have what's called the alpha helix, and we also have what's called a beta-pleated sheet. The alpha helix when we look at this one, the structure for this is maintained by hydrogen bonding interactions between the NH and CO groups along the peptide backbone of the coiled protein strand. So what will happen here is the amino acids line up so that you get a hydrogen, this hydrogen bonding between the hydrogen off of this nitrogen and the oxygen of the carboxyl group. So we have hydrogen bonding here. So these line up in a coil, and you get those interactions. So you have intermolecular forces coming into play here. With the beta-pleated sheet, here what happens is they line up slightly differently. And so the beta-pleated sheet is maintained by interactions between the peptide backbones of neighboring protein strands. So in this structure, the chain is extended as opposed to coil. So rather than having it in a coil, it kind of looks like sheets of paper that are kind of stacked on top of each other. And this forms a zigzag pattern like an accordion plate. So here you get interactions between the different peptides. All right, so what this does, so some proteins such as keratin, which composes hair, has the alpha helix pattern throughout their entire chain. So like our hair, we know it's a protein. Why is it that you're able, like those of you who have curly hair, and you can use an iron to get the hair to straighten, why can that happen? What's happening to your hair when you do that? Any ideas?
Are you breaking those? Bonds?
You're interfering with those hydrogen bonds. Yeah, that heat gets in the way, and you denature the protein. That's what happens. This particular process is reversible because as soon as you wet it with water, what happens? Goes back curly again, right? So those hydrogen bonds are going to come back, and you get that curly hair again. Temporary denaturing. When you cook eggs, obviously that's like a permanent denaturing. You can't reverse that process. So sometimes denaturing proteins is permanent, sometimes it's not. Sometimes it's semi-permanent, like when you use chemicals for, I don't know, bleaching or not bleaching. Can you use chemicals to straighten hair?
Yeah.
I'm not a
like you
beautician,
get like
so.
They do perm.
the blowout.
Yeah, and you do perm, so it's like semi-permanent. They eventually leave, but they're there for a temporary basis. Okay. Some proteins, such as silk, have that beta-pleated sheet structure throughout their entire chain. Since its protein chains in the beta-pleated sheet are fully extended, silk is inelastic. So silk, you can't really, it's not like you can iron it and stretch it and pull it, whereas like your hair, you can kind of pull it in a little bit because it has, once again, that alpha helix pattern. Many proteins have some sections that are beta-pleated, others that are alpha, and still others have sections that are less regular patterns called random coils. Okay, that's why it gets longer when it's wet, basically kind of what we just talked about too. Yeah, and that's another reason why when you get your hair cut, they should blow dry it at the end to make sure it's actually straight. All right, tertiary protein structure. It consists of the large-scale bends and folds due to interactions between the R groups of amino acids that are separated by large distances in the linear sequence of the protein chain. So interactions for this one, there's a few different types of interactions that you need to be aware of for tertiary protein structures. Hydrogen bonds, disulfide linkages, and these are covalent bonds between sulfur atoms on different R groups. Hydrophobic interactions, those are attractions between large nonpolar groups, and then salt bridges, which is an acid-base interaction between acidic and basic groups. Proteins with structural functions such as keratin, which compose hair again, or collagens, which compose tendons and much of the skin, tend to have tertiary structures in which the coiled amino acid chains align parallel to one another, forming long, water-insoluble fibers, hence the reason your hair doesn't dissolve in water. These kinds of proteins are called fibrous proteins. Proteins with non-structural functions, such as hemoglobin, which carries oxygen, or lysosome, which fights infections, tend to have tertiary structures in which the amino acid chains fold in on themselves, forming water-soluble globules that can travel through the bloodstream. These kinds of proteins are called globular proteins. Many proteins are composed of more than one. Is that all I do on that one? Wow, that is. Okay. Many proteins are composed of more than one amino acid chain. This is going to be for a quaternary structure. Recall that hemoglobin is composed of 4 amino acid chains. Each chain is called a subunit. The quaternary protein, sorry, I hit a key without meaning to. Where did I just go? I went all the way to the end. I hit end. Darn it. Whee! Let's find out where we are.
There. Okay.
Did I read this already? No.
Right.
Okay.
Oh,
The quaternary protein structure describes how those subunits fit together. The same kinds of interactions between amino acids maintain quaternary structure and tertiary structure. So you still have your hydrogen bonds, your hydrophilic, your salt bridges, and your disulfide. Here we go. This is what I was looking for. Okay, so for the tertiary and quaternary interactions, we said there's 4 basic interactions. Up here at the top, you see our hydrogen bonding. Remember, hydrogen bonds can only form between hydrogen and what 3 elements?
Nitrogen, oxygen, and fluorine.
Nitrogen, oxygen, and fluorine. So that's the hydrogen bonding. Up there, you have between oxygen and hydrogen. We have a hydrophobic interaction down here. You have these long hydrocarbon chains. Over here is our disulfide linkage. You have 2 sulfurs, and cysteine is one of the only ones that can actually do that. The disulfide linkages don't happen with very many amino acids because they have to have a sulfur.
Wait, so the shape that it takes on is because of the bonds? It's kind of like a...
Right, so what happens is, like, you have this long protein backbone, this protein chain, and you'll have serine over here, and I think this is threonine, and what will happen is there'll be an attraction between that hydrogen and oxygen so that folds up so that they can actually come up into that interaction with each other.
Okay.
Same thing here. There's a connection here between these 2 bonds, so there's a slight fold here. Same thing with this. There's a connection between those 2 sulfurs, so there's a slight contraction right there. Down here we have a salt bridge between glycine and arginine, and so they come in and they kind of, once again, come in proximity. So that's what's responsible for all those folds and the shapes that it actually takes.
And those are the acid-base ones, right? Because that's...
Salt bridge is the acid-base, yeah.
Okay. Trying to follow along.
I know, it's a lot to take in. Okay, so to summarize the protein structure, the primary structure is the amino acid sequence. Secondary structure refers to those small-scale repeating units and the patterns found in the protein. So we had our alpha helix and our beta-pleated sheets for those ones. Our tertiary structure is those large-scale twists and folds. So that was our hydrogen bonding, our hydrophobic, our disulfide bonds, and our salt bridge interaction.
Are those the same as random? The random fold or something like that?
The random was part of the secondary. So those ones aren't as common. We mainly focus on the alpha and the beta-pleated. Okay. The quaternary structure was the arrangement of the chains or the subunits in proteins, which is also maintained by those same 4 interactions, the hydrogen bonding, disulfide linkages, salt bridge, and your hydrophobic ones. Okay, how are we doing so far? It's a lot to take in, yeah. Hence the reason it's multiple choice test. Okay, nucleic acids. This one, hopefully, is a little bit more familiar with most of y'all because I think there's a pretty big emphasis on this in high school sciences. So when we're looking at the final class of chemicals in our bodies, we're looking at nucleic acids. And these are going to ensure that our proteins have the correct amino acid synthesis sequence. Nucleic acids contain a chemical code that specifies the correct amino acid sequences for the proteins. And they can be divided into 2 types, our deoxyribonucleic acid, or DNA, which exists primarily in the nucleus of the cell, and ribonucleic acid, or RNA, which is found throughout the entire interior of the cell. Like proteins, nucleic acids are polymers. The individual units composing nucleic acids are called nucleotides. Each nucleotide has 3 parts, a phosphate, a sugar, and a base. In DNA, the sugar is deoxyribose, whereas in RNA, the sugar is ribose. So hence the difference between DNA and RNA. You have deoxyribose versus ribose. Deoxy, you have no oxygen. Ribose, you have the oxygen right here. So the components of DNA. DNA, once again, it's a polymer nucleotide, and each nucleotide has the 3 parts. We have our sugar group, our phosphate group, and a base. So the DNA is going to be a long chain where the phosphates are connected to a sugar, and there's a base sticking off to the side of them. So you have phosphate, sugar, phosphate, sugar, phosphate, sugar, goes all the way down the chain. The phosphates, just a phosphate group, always the same thing, this P with 4 O's attached to it. And then the sugar in this case, because this is DNA, it's going to be deoxyribose. And then you have your base sticking out to the side. The bases, that's your 4 different bases that you've learned about. Your A, you can see the A, T, C, and then also G, which is not on there. So the nucleotides are joined by phosphate linkages from your phosphates that are on there. Every nucleotide in DNA has the same phosphate and sugar group, but can have one of 4 different bases. So we have our A, C, G, and T that you've learned about, and you probably did all those little games with the A to C and G to T and that kind of thing. And RNA is basically the same, except for the sugar is different. It's the ribose instead of deoxyribose. And the base uracil replaces thymine. So you have just uracil instead of the T. The order of bases in a nucleic acid chain specifies the order of the amino acids in the protein that it's coding for. Since there are only 4 bases and about 20 different amino acids to be specified, a single base cannot code for a single amino acid. It takes a sequence of 3 bases called a codon to code for 1 amino acid. The genetic code, which is the understanding of which amino acid is coded for by which specific codon, was discovered in 1961. It is nearly universal. The same codons specify the same amino acids in nearly all organisms. In DNA, the sequence AGT codes for the amino acid serine, and the sequence TGA codes for the amino acid threonine. So just that reversal gives you a whole different amino acid. In a rat, a bacterium, or a human, the codes tends to be the same. Okay, so kind of break that down into a pictorial representation here. So here is our DNA. We have, this is our sugar phosphate backbone right here, and these are the different bases that are on that particular DNA molecule. 3 bases form together to form a codon. This tells the body what protein or what amino acid to actually synthesize. So you have 3 bases, form your codon, now you know which amino acid to actually form. A gene is a sequence of codons within a DNA molecule that codes for a single protein. Because proteins vary in size from 50 to thousands of amino acids, genes vary in length from 50 to thousands of codons. Each codon is like a 3-letter word that signifies 1 amino acid. String the correct number of codons together in the correct sequence, and you have a gene, which is the instructions for the amino acid sequence in a protein. Genes are contained in structures called chromosomes. We have 46 of those within the nuclei of the cells. So organization of the genetic material in us, we have our chromosome up at the top, which is a macromolecule of DNA. What does macromolecule mean?
Small.
Macro. It's huge. Macro is big. Micro would be small. So it's a macromolecule. It's a very large DNA molecule. Then within that chromosome, you see that we have genes, which are a portion of the DNA that codes for a single protein. So in those genes, you have codons, which are that sequence of 3 nucleotides and their associated bases, and that codes for 1 amino acid. Then within that codon, you have the 3 bases, pull one of those down, and you have your nucleotide, which is that link in the nucleic acid chain. Okay, so DNA structure. If we look at the structure of DNA, we all know that alpha helix structure that they always show, they always make it look really pretty. The ability of DNA to copy itself is related to the alpha helix structure. DNA is stored in the nucleus as a double-stranded helix, and the bases on each DNA strand are directed toward the interior of the helix, where they hydrogen bond to bases on the other strand. So once again, intermolecular forces are playing a huge role in our bodies. The hydrogen bonding between the bases is not random. So this is where we get that A to T, G to C thing. Each base is complementary, capable of precise pairing with only one other base. Adenine will only hydrogen bond with thymine, and cytosine will only hydrogen bond with guanine. So it's A to T, G to C. When a cell is about to divide, the DNA within the nucleus unwinds and the hydrogen bond joins the complementary bases. They break, forming 2 single parent strands. So your DNA, the hydrogen bonds break, splits into 2 different strands. With the help of enzymes, a daughter strand, which is the complementary strand to each parent, is formed. And then the hydrogen bonds between those strands then reform, resulting in 2 complete copies of the original DNA, 1 for each daughter cell. So now, instead of 1 DNA, we have actually 2 that are identical to what we started with. All right, so going back to high school biology here. What is the sequence of complementary strands for the following DNA? So if you have A, what does it pair to?
T.
T. What does the T pair to? A, G,
C.
A,
T.
G, T, A, and C.
G.
Okay, that's on the exam. Don't miss that one. I don't know if it's this exact question, but something like that. Okay, let's see here. I think we got that. All right, when we look at the nucleic acids, when we look at humans and animals specifically, we must synthesize the proteins that we need to survive from our dietary proteins that we eat. That dietary protein is split into its constituent amino acids during digestion, and these amino acids are reconstructed into the correct proteins, those needed by the particular organism in the organism's cells. And nucleic acids will direct these processes. You've probably heard of essential amino acids and non-essential amino acids. Have you all heard those terms thrown out before? Essential and non-essential amino acids?
Kind of.
What is the difference between them? What do you think non-essential amino acids are?
We don't do that when it's important.
Do we not need them?
No.
That's not what it means. It means that our bodies actually synthesize those amino acids. So non-essential amino acids are ones that we do not have to eat. Our body can make them. Essential amino acids, our body cannot make. We have to get them from our diet. So there's essential amino acids that you have to actually go out, make sure you're eating an appropriate diet so that you can build the proteins that your body's supposed to build. If you don't have enough of the essential amino acids, your body can't make those proteins. So that's kind of the necessity of them. When a cell needs to make a particular protein, the gene, the section of the DNA that codes for that specific protein unravels. That segment of DNA corresponding to the gene acts as a template for the synthesis of a complementary copy of that gene in the form of another kind of nucleic acid, which is the messenger RNA. So you probably remember seeing this mRNA stuff. That's what we're talking about here. The mRNA moves out of the cell's nucleus to a cell structure within the cytoplasm called a ribosome. And at the ribosome, that's where the protein synthesis actually occurs. The mRNA chain that codes for the protein moves through the ribosome, and as the ribosome reads each codon, the corresponding amino acid is brought into place, and a peptide bond forms with the previous amino acid. As the mRNA moves through the ribosome, the protein is formed. Okay, so this is kind of a pictorial representation as to what we're talking about here. So here we have the messenger RNA. The messenger RNA is this long strand that comes from our DNA. We've now coded it for the mRNA. And it comes into contact with a ribosome. Ribosomes have a large subunit and a small subunit, and the mRNA fits kind of in between them. So this mRNA, the ribosome travels along the mRNA, and as it's doing that, it reads the sequence of those bases. So every 3 bases, it knows what protein or what amino acid to code for. So the amino acids are built, and it keeps going down, and it keeps growing those. The secondary structure begins to form as it gets longer. Then you're going to eventually have this complete protein with the tertiary and the quaternary structure at the very end.
Wait, so it's like printing the information, kind of?
It's kind of like printing, yeah. It's kind of like it's translating what's being fed to it.
And after it does that, does the mRNA does it keep doing that, or does it have to go back?
Well, eventually it'll be told stop. There's a stop codon, and so it'll stop doing stuff. There's I don't know that I have a slide up here that tells us what the codons are, but there's stop codons and there's start codons that it tells the mRNA when to start copying it and when to stop copying it. Yeah, our bodies are amazing machines.
Yeah.
Okay, so to summarize this, DNA contains the code for the sequence of amino acids in proteins. A codon, which is 3 nucleotides with their bases, codes for 1 amino acid. DNA strands are composed of 4 bases, each of which is complementary, which is capable of precise pairing with only one other base. A gene is a sequence of codons, which codes for 1 protein. And chromosomes are molecules of DNA found in the nuclei of cells. Humans have 46 chromosomes. When a cell divides, each daughter cell receives a complete copy of the DNA, all 46 chromosomes in humans within the parent cell's nucleus. When a cell synthesizes a protein, the base sequence of the gene that codes for that protein is transferred to mRNA, and the mRNA then moves out to the ribosome where the amino acids are linked in the correct sequence to synthesize the protein. So the general sequence to complete a protein is you have your DNA, which goes to the RNA, which goes to the protein. Okay, so drugs for diabetes, we talked about that a little bit with the insulin. So diabetes is that disease in which a person's body doesn't make enough insulin, which is a substance that promotes the absorption of sugar from the blood. Consequently, diabetics have high blood sugar levels, which can over time lead to a number of complications, which includes your kidney failure, heart attacks, strokes, blindness, and also nerve damage. One treatment for that, which is the most common treatment, is the injection of insulin, which can help manage those blood sugar levels and reduce the risk of those complications. Insulin is a human protein that cannot be easily synthesized in the laboratory. So for many years, what they would do is they would use the insulin from pigs and cattle. And although that animal insulin worked to lower the blood sugar levels in some of them, it couldn't in others. So some patients, they couldn't use it. Today, they actually use human insulin. They were actually able to replicate that in a lab. So scientists were able to remove the gene for insulin from a sample of healthy human cells, and then they insert that gene into a bacteria, which incorporated the gene into their genome. This is genetic engineering, by the way. So not all genetic engineering means cloning. There's some actually decent forms out there as well. So no, when people talk about genetic engineering, it's not always all bad. Some of it's kind of scary, it's not all. When the bacteria reproduced, they passed on exact copies of the gene to their offspring, and this result was a colony of bacteria that contains the human insulin gene. Then the chemical machinery within the bacteria expressed the gene, meaning that the bacteria synthesized the human insulin that the gene codes for. Today, insulin made in this way is harvested from the cell cultures and then bottled for distribution to diabetics. So they actually get human insulin, and there's a lot less chance for rejection that way. Okay, that's chapter 19. Piece of cake, right?
Oh yeah, ready for the test.